Hello. Thank you for paying attention to our work published in Cell Regeneration about activation of the melanocortin 4 receptor signaling in digital regeneration. I'm Gu-Fa Lin. I'm pleased to walk you through this short presentation. And thank you for your time, and I hope you enjoy it. Our lab works on limb regeneration. In addition to axolotls, we use Xenopus laevis as a model system. The reason we choose Xenopus laevis is because Xenopus, like axolotls, can regenerate the limb. But unlike axolotl, Xenopus can only regenerate the limb at a very young tadpole stage. So a stage 53 tadpole can fully regenerate the hind limb, but a stage 58 tadpole cannot regenerate the limb. We also use a mouse digit as a model system to study regeneration. That's because the mouse digit can actually regenerate the terminal tip. So if less than 50% of the terminal phalange, P3, is amputated, it can regenerate. This is the P3 distal amputation. However, if proximal part of the P3 is amputated, it will not regenerate. And any level closer to that, to the body, including the P2 amputation, will not regenerate. So the Xenopus has an age-dependent regeneration capacity in the limb, and the mouse has a level-dependent regeneration phenomena in the digit. These model systems provide us a very good platform for try-out method to stimulate regeneration. And in the last couple of years, we have looked at it various aspects of limb regeneration and digital regeneration, including the signaling pathways and the possibility of using progenitor cell transplantation together with growth factor treatment to stimulate regeneration in the mouse and Xenopus. And more recently, we have looked at the nutritional supply, the energy metabolic homeostasis in limb regeneration. In mammals, food intake and energy homeostasis is tightly regulated by the central hypothalamic melanocortin pathway, and the key player of this pathway is the melanocortin 4 receptor, Mc4r. The tadpole also has a hypothalamus, of course, but there are not many studies on energy regulation in the amphibians. So we started to look at the energy regulation in Xenopus, and we asked the question about whether hypothalamus has any function in regeneration, like limb regeneration. So we decided to examine limb regeneration after hypothalamus injury. Here is the area of hypothalamus in the Xenopus tadpoles. We used electrocautery to damage the hypothalamus, and we found that the tadpole do not regenerate the limb anymore. So we can see that the limb is very pale in color. That is because the level of α-MSH in the tadpole limb is significantly reduced when the hypothalamus is injured. So this shows that central hypothalamic signaling is required for tadpole limb regeneration. So we know there are 5 receptors in the melanocortin systems. So we further went to identify which one is involved. As we can see here, Mc4r is the melanocortin receptor that functions in the limb during development and during limb regeneration, and its expression is downregulated if the hypothalamus is injured. And its loss of function by morpholino knockdown also inhibits limb regeneration. So Mc4r signaling regulates limb regeneration in the Xenopus tadpole. So the questions we want to further address in this cell regeneration paper is about the Mc4r expression in the mouse, limb, and digit, and what is the function of Mc4r signaling in digit regeneration. When we look at the expression of Mc4r in the Xenopus tadpole limb, we observed that Mc4r is closely co-localized with limb nerves, which raises the possibility that Mc4r signaling may have a neurotrophic role in limb and tadpole regeneration. For the expression of Mc4r, we use the Mc4r GLP transgenic mice that we obtained from Dr. Hu-Xin Cui's lab, and we examined the Mc4r GLP expression in the developing limb from E10.5
to neonate and adult. There are more details in the paper, and I encourage you to read it. But the basic idea is that, first of all, the expression of Mc4r is mainly related to nerve tissues in the limb. And we also observed that Mc4r is expressed in the muscle tissues when the limb nerve started to innervate the limb muscle, which corresponds to a secondary myogenesis process in the limb and occurred at about E13.5. More interestingly, we noted that expression of Mc4r become increasingly restricted in the nail organ in the neonate and adult mouse, and after digit amputation, its expression is upregulated in the blastema. And in the Mc4r mutant mice, we found that the digit regeneration failed to occur. More interestingly, than the Mc4r heterozygous mice, the digit can still regenerate, but it is very seriously delayed. So, to answer the question about the function of Mc4r signaling in the mouse, we first utilized this Mc4r heterozygous digit and to examine the effect of α-MSH supplementation to the mouse to see whether there is any rescue effect of activating Mc4r signaling on the mouse digit. As we can see here, α-MSH rescued the delayed digital regeneration in the Mc4r heterozygous, and to an extent as equivalent to the wild-type controls. And the second experiment we did was to supplement α-MSH to the mouse digit when the mouse is denervated. So we know that denervation of the hind limb will inhibit regeneration of the digit tip. However, when we inject α-MSH in these mice, denervated mice, we can see that the digit tip has been restored. And more interestingly, we can see that there are a nail organ formation in these Mc4r GLP transgenic mice, suggesting that the α-MSH supplementation can rescue the denervated digit in digital tip regeneration and has a function in the nail organ formation. So the third experiment we did was to examine whether α-MSH supplementation has any stimulating effect on proximal digit regeneration. As we know that the P3 proximal amputation will not regenerate. However, when we inject α-MSH in these mice, we observed that there are significant digital regeneration rescued. And when we look at the area of this regeneration, it shows that there are very obvious difference. The result shows that Mc4r is expressed in the mouse limb, and Mc4r expression is closely related to the nerve tissues, and α-MSH Mc4r signaling has a neurotrophic function in the mouse digital tip regeneration as well. This suggests that α-MSH Mc4r signaling can be further explored to stimulate proximal digital regeneration. And indeed, the indication to our work is that we can utilize this model system and combine the activation of this signaling pathway together with a method of generating limb progenitor cells from stem cells in 3D culture and combine them together to make transplantations to proximal amputated limb and digit to achieve better regeneration in the mouse. That concludes my presentation, and we are very grateful for our collaborators and the funding agencies. And again, thank you very much for your attention at the time. I hope you continue to pay attention to the content of cell regeneration.